The title of this presentation is Misclassification Errors of Machine Learning Pipelines and Electronic Structure Prediction of COVID-19 and CellTalkDB Receptors. To begin with a presentation overview, we'll be introducing machine learning and deep learning algorithms for compound discovery and enzyme design. And we'll have to motivate connections between quantum algorithms, molecular docking, and silico simulations so that we can describe how previous implementations of open source machine learning frameworks, which can be compared with the output of deep learning workflows, can enable us to discuss as somewhat of a high level of generality components of the classical molecular docking workflows which share in characteristics of quantum molecular docking workflows. And this is to say that in previous videos of implementing molecular docking simulations, we have investigated novel compounds with Autodoc Vina before, which suggests that this is a good point of comparison to establish between quantum and classical algorithms, in which we can determine whether there's any difference in the expected runtime of different algorithms when looking at running molecular docking simulations. And in this previous video, I looked at the CellTalkDB database, which is a publicly maintained repository from scientists in China, in which they manually group together ligand and receptor pairs, which I'll describe further in depth. And this is very interesting because it establishes whether the Autodoc Vina or other open source machine learning workflows can be used to characterize molecular binding affinities in a different conformation, such as for the COVID-19 receptor, in which from a reference on protease inhibition that I discussed in another video on the channel, I looked at making use of and establishing comparisons between machine learning workflows and deep learning workflows and predicting the binding affinity across different pockets that are dispersed throughout the surface of the receptor for COVID-19. In this second paper reference, highlights our attention to the fact that there are other possible databases for discussing SARS-CoV-2 drug candidates, which are related to the production or the inhibition of protease that is produced by the coronavirus. In this second reference, the authors draw our attention to the fact that they can make use of Autodoc Vina, which is the same open source framework that I discussed in a previous discussion or presentation on the CellTalkDB database, in which they are able to generate 4 different possible conformations which interact with the COVID-19 receptor in the following ways. So they're able to determine whether there's any high probability that any one of these conformations can interact strongly with the COVID receptor, which could have implications for the ways in which not only the COVID receptor, but other disease receptors for respiratory diseases or other types of complications can be propagated within a host organism from the initial infection. And on the other hand, the CellTalkDB database is a public repository maintained by scientists in the Chinese Academy of Sciences, in which they look to form different ligand receptor pairs, abbreviated LR, for the human organism as well as the mouse model organism, and to determine whether there are any differences in how diseases or ligand and receptor pairs can interact with one another. And it's very much helpful to have any type of information available from the mouse model organism, because it allows us to determine what are the differences in different modes and complexities of propagation of diseases in mouses and compared to humans, as well as determining any of the simpler interactions in the humans and whether this interaction between ligands and receptors can be modeled in how diseases propagate and manifest in mouse model organisms. And the CellTalkDB GitHub provides an overview of how the data collection and data processing stages is implemented for determining which ligand receptor pairs are helpful to have in mind when running molecular docking experiments or simulations in silico. For the in silico simulations, we need to look at the fact that there are different protein-protein interactions, as well as other annotation information, which then needs to be processed to data processing stage, which consists of removing any duplicate protein-protein interactions, as well as characterizing other ligands and receptors which could be of use for further examination in determining whether any ligand and receptor pairs interact strongly with each other. But besides public repositories which have previously been examined, it is possible to push the boundaries of molecular docking workflows with compounds whose binding affinities are difficult to characterize experimentally. And the benefits of examining such compounds with in silico methods include the 3 points that I've given here that I'll reiterate, but there are also many more. The 3 points, which are some of the easier ones that come to my mind when I'm thinking about these topics, is that we can obtain approximations of binding affinity for compounds from simulations before testing compounds manually with experiments. So not having to look into any type of sophisticated assay development or really any type of assay development at all, because in the molecular docking simulations, we're able to draw information immediately from the PDB file, which is publicly available information, and then we're able to pass search coordinates, which define a search volume, which I'll reiterate and further discuss with the algorithm that is able to immediately process whether any conformations will be likely to materialize between some ligand and receptor. And we can also look at an advantage in terms of being able to run a polynomial time algorithm for discovering enzymes within an exponentially large search space that performs a desired chemical reaction optimally. And finally, for the third bullet point, we can express several candidate compounds simultaneously by running all the simulations in parallel. And as mentioned in another presentation on the channel, the one that focused on protease inhibition, it is of utmost importance to compare binding affinity conformations across several different virus structures for determining how replication events occur within host organisms. And this is very much related to the fact that there is a high degree of genetic similarity between the HIV virus and SARS-CoV-2. And we can even look at for the HIV virus, there are several different types of models which are available, which have different type of visual representations and which occupy space differently. We can see that there are these two type loops, overlapping loops, which appear to intersect around the middle of each one of these possible HIV models. But we can see that the loops on either side of this possible intersection point, they fold differently, and they actually, which can pose different thermodynamic consequences for how ligands or smaller compounds can interact with different areas of the HIV receptor. And from several candidates for the model HIV virus, as we have been discussing in the previous slide, the couple of models below exhibit significant differences in the region of the surface of the receptor that can initiate a replication event for the virus. And I'm just providing specific arrows to the types of differences in the structure and the morphology of the receptor that I mentioned in the previous slide, in which there can exist some different ways in which either one of these loop-like structures of the HIV virus from several different publicly available models can fold onto one another, which can have different types of implications for how smaller compounds or other molecular vesicles within the human body would interact with this receptor, which could result in an infection event in which the virus begins to replicate and infect the host organism with its genetic material and changes the host organism's genome in the process and genetic material. And to prevent unwanted errors from molecular docking workflows, we first need to describe all the parameters that are passed to the algorithm, which we can then use to describe what is a misclassification error of a machine learning or other types of AI or deep learning pipelines, and how one may be able to avoid this type of error. And this is given by the fact that if you want to run AutoDocVina, which is one of the open source molecular docking workflows, we have to specify a receptor and a ligand in a PDB-QT format, as well as specifying the X, Y, and Z centers, as well as the size of the X, Y, and Z dimensions that we would like to search over, as well as finally the PDB-QT output, which will contain some countable number of conformations that we would like to look into further. And after passing all the necessary parameters to AutoDocVina, one possible molecular docking open source framework, a list of conformations of the final output file is produced. And we can see that from some previous iterations of the molecular docking workflow that ran for AutoDocVina, which one can also run for other molecular docking workflows, is that there's some finite number of modes which correspond to individual conformations that the algorithm predicts with high probability could occur, as well as the corresponding binding affinity in kilocalories per mole. And the other two items which are given in the column are looking at different ways in which the distance of the ligand and receptor can vary, which can result in different geometries about a binding pocket that can affect or that can impact the types of conformations and predictions that we can generate from running this in silico simulation. But unfortunately, sources of error can accumulate in molecular docking workflows if a poor choice of parameters for the docking regions are passed to the algorithm. And I was able to see more of these errors propagate in a more significant way from some receptors in the CellTalkDB database that I introduced in the previous slide, because from that collection of ligands and receptors, we can see that conformations that are produced by the algorithm in the PDB-QT output can be oriented directly outwards of the binding pocket or in other directions which are not directly lining up with the main component of interaction that we would expect to see between a ligand and receptor of interest in a binding pocket. And this is important because this is not the type of interaction that we would like to experimentally characterize with a binding assay that we would say like to pass on to our experimental colleagues or other types of colleagues that are more away from the data science or computational biology side of things and actually want to compare and develop and design different experimental assays or protein binding assays or various kinds of assays which can experimentally confer the conformations that we're able to view the geometry of and study from our in silico molecular docking simulations or experiments. And additional sources of error from the conformations produced from the molecular docking algorithms can also result in incorrect interpretations of complex formation after binding to a receptor of interest has occurred. And this is very much, again, related to part of the HIV virus and the high percentage of genetic similarity that are, say, shared between SARS-CoV-2 and HIV, because HIV, it begins with, as we can see in the leftmost image here, it begins with a maturation stage in which the protease inhibitor plays a role in preventing or in the infection and the replication stage, in which there can be some assembly mechanism in which the virus, if it doesn't have the protease inhibitor, it won't be able to undergo a maturation stage. However, in the presence of the protease inhibition, the HIV proteins, which are ultimately responsible from the CD4 cells for eventually propagating the virus, they're able to develop protease, which is what distinguishes a mature from an immature HIV cell or body, which will then allow the mature HIV to propagate the infection of genetic material. So this is just demonstrating that the final image is demonstrating that once the CD4 cells reach maturity, the HIV proteins are able to, in the presence of protease, they're able to stop the new HIV from becoming mature HIV. So it's demonstrating different types of mechanisms that are associated with disease receptors beyond just the binding affinity or the conformation itself, which are very important and can be misguided if we don't have the appropriate conformation with the right orientation into the binding pocket of any receptor that we're looking at. And similar problems exist for hundreds of receptors that are gathered in the CellTalkDB repository. So it's very much still appearing for a while, at least, that it's going to be of interest to apply different types of algorithms and to vary the parameters of these machine learning, deep learning, or even quantum algorithms for determining how misclassification errors in the conformations that are produced from the PDB-QT output files can be avoided. And such problematic configurations produced in or conformations produced in the PDB-Q output files prevent machine learning pipelines from being deployed without having the conformations individually checked for accuracy. Because if we have some lipid conformations in different modes, like how we demonstrated can even be produced from the off-the-shelf machine learning workflow or pipelines from molecular docking simulations, we can see that we would have to examine all these 9 conformations to apply different types of checks, namely very common sense checks, namely determining what is the orientation of the ligand and the receptor relative to each other, which can then help us determine whether a conformation that's produced from the in silico simulation is actually something that could appear in a wild type interaction that would then be responsible for changing the genetic material of the host organism that is being infected and being attacked by the virus. In diagnosing possible sources of error and problematic conformations from the PDB-Q output can include asking ourselves, was the search region not of the appropriate size? Was it either too small or too large? Or are the binding pockets within the designed search volume, as well as from the other side, from the other direction, is the predicted binding affinity from the compound structure too weak in the kilocalorie per mole scale that's output, which are the units of the binding affinity that are output by AutoDocVina and other open source molecular docking workflows, as well as asking ourselves if similar compounds have been tested in assays before. So the concluding remarks that I want to finish up with are that we introduced machine learning and deep learning algorithms for compound discovery and enzyme design, and we discussed computational difficulties in predicting properties of electronic structures of compounds, which can raise difficulties for predicting binding affinity, determining the characteristics of smaller compounds interacting with a receptor of interest, as well as directed evolution of enzymes. And we drew attention to previous problematic conformations produced by the open source molecular docking workflows, with attention to future areas of research and improving prediction capabilities of new algorithms. And thanks for watching.